Okay, we've got to get moving with today's lecture. So, luckily, lecture and lab are coinciding once again. So we're going to start digestive. You already saw a lot of these parts in lab last time. So you should be able to tell me a lot of this beginning stuff. Our goal for today, we're not doing the chapter in the same order that the chapter goes. Okay, we're going to jump around a little bit. And the reason for that is, well, before we can really understand this functional anatomy stuff here in part 2, we've got to understand the physiology of how we break down food stuff. Okay, so we're going to look at part 1 and part 3 today. And I'll try to keep it as simple as I can, but it is going to be a fair amount of chemistry this morning. So, yeah, nothing like starting out the day that way, right? Also, I know on your syllabus it said we were only going to take 2 days for digestive and then 2 days for metabolism. This book arranges things a little bit differently, so we're going to switch that. We're going to have 3 days for digestive and 1 day for the metabolism and nutrition stuff. All right, so I have the digestive stuff laid out for you here. We'll wrap up the digestive stuff next week. So how do we organize the organs in this system? We have 2 major groupings. We've talked about groupings of organs in other systems, so this shouldn't be a surprising way to start out. We group organs as either being part of the alimentary canal, which we can also call the gastrointestinal tract or GI tract, or we can classify organs in this system as accessory. So when we talk about the ones that are part of this alimentary canal, these are all of the organs that are going to hold the food stuff at some point. And you guys already know all of these because we talked about them last time in lab. So where does food enter the body? What's our fancy term for the mouth? Oral cavity. That's right. Okay, so we've got the oral cavity. And then where does the food hit next? The pharynx, specifically the oropharynx. From there, where does it go? Whoa, from the oropharynx to the laryngopharynx, and then the esophagus. And then from the esophagus, where's it headed? To the stomach, exactly. And then from the stomach, where does it go? Small intestine. And then to the large intestine. Didn't we talk about regions of the intestines in lab last time? Yeah, we did. How many regions are there in the small intestine? Only 3. What are they? Starting at the first part that the food is going to hit. Then we have the duodenum, then the jejunum, and the ileum. Good. From the ileum, it's going to hit the large intestine. What specific region? It's one of the few parts that's not a colon. It starts with a C. The cecum, exactly. So it hits the cecum first, and then from the cecum, what part of the colon does it enter? Ascending, good, and then transverse colon, and then descending colon, and then it hits the part that has the curves like an S, right? So what's that? The sigmoid, and then it hits the rectum, and then the anus. All right, and I have all of those things highlighted for you on this screen. So it's a good idea to just go back and review them. Accessory organs don't hold the food stuff, but they help out with digestion. So these are going to be things that, a lot of them anyway, are going to be things that secrete stuff that's going to help us to break down food stuff. So notice, a lot of the things that I'm listing out here are glands, right? And we know glands secrete things like the salivary glands that we studied in lab last time, pancreas, you know that's a gland. It's going to be helping us here in the digestive system. So is the liver. Make a note now, because this is going to keep coming up through this chapter. The liver and the pancreas, when they secrete stuff, they secrete it specifically to the start of the small intestine. Where was that? The duodenum, good. Duodenum, duodenum, tomato, tomato. The other stuff that I have listed isn't going to secrete, but it still helps out. So when the liver makes its secretion, which that secretion is going to be called bile, the bile actually gets stored in the gallbladder, and it isn't just a storage container. It pulls water out of the bile so that it becomes more and more concentrated. Teeth and tongue obviously are going to help us with some physical breakdown of the food stuff, right? Like when we chew, the teeth are mashing food stuff up. So if we look back at that figure, we can see a lot of those accessory things listed here as well. Liver and pancreas we'll be talking about more in lab today, since we didn't talk about them in lab last time. Just like we talked about with the respiratory system, remember how we said the respiratory system happens because of several processes? Digestion is going to happen due to several processes as well, except even more. We have 6 processes that help us with digestion. Well, you can't exactly digest and break down the food if you don't bring it into the body, so it should make sense that the first thing is ingestion, bringing the food in the body through the oral cavity. We have to move the food through this tubing of the alimentary canal or GI tract, so we're going to call that movement propulsion. And I have 2 specific examples on the slide. Notice I have swallowing and peristalsis. Swallowing is something we're going to talk about more next time. Our fancy term for it is deglutition. And that's going to involve both skeletal muscle and smooth muscle, because think about it, when you decide it's time to swallow your food, that's a conscious thought. You start that process with skeletal muscle, which you voluntarily control. But then, as we move the food through the esophagus is going to turn to smooth muscle in the wall, so that we don't have to consciously think about squeezing the food all the way down to the stomach. When they talk about peristalsis here, this is a term that we're going to see again, so make sure you learn it well now. What that means is that the muscle in a tube, like the smooth muscle in the esophagus or the small intestine, it's going to sequentially squeeze the food so that it's squeezing toward the opposite end of the tubing. So when I think about peristalsis, I compare the GI tract to a tube of toothpaste. You know, when you have that last little bit of toothpaste left in the tube and it's stuck all the way on the end opposite of the opening and you have to squeeze it all the way along the tube to get it out, that's kind of what peristalsis does for us. When it comes to the actual breakdown of the food, we have 2 different types of digestion. We have mechanical digestion and chemical digestion. With the mechanical digestion, well, anytime we see mechanical, we know that that means something physical is happening, right? So with the mechanical digestion, a good example of that is chewing. That's not the only place where we see mechanical digestion happening, though. We're going to see it in the stomach. We're also going to see it in the small intestine. And specifically, when we talk about it in the small intestine, that type of contraction is going to be segmentation. So that's a word in bold here. And even though I'm really emphasizing that this is going to help to kind of mash up the food, this is also going to help us to mix up the food stuff with the chemicals that we're going to be adding to it. And that's really what segmentation does for us. if we look back at our figure, there was another half to it. And let's notice what's going on here. They're color coding the food stuff so that we can see it's kind of being compartmentalized here in the small intestine, right? So as we contract the smooth muscle here in the middle of this piece, that's going to force some of the food stuff backward and some of the food stuff forward. That way we can make sure that everything gets mixed up with the chemicals that are going to help us break it down. So that's kind of the purpose of segmentation. Well, I mentioned chemicals, so it shouldn't be surprising that the other part of digestion is chemical digestion. And specifically with this, we are going to be focusing on enzymes. Enzymes that are going to break the chemical bonds that hold the nutritional molecules together. Because we have to break them down to a small enough size so that our body's cells can absorb those nutrients. In order to get the nutrients in the body, we call that absorption. Make a note that most of that happens in the small intestine, but we will see a little bit in the stomach as well as in the large intestine.
The absorption, it's always the same, or it depends on many factors? I mean, you are going to absorb the same quantity, or it's going to be like there's other factors that could influence absorption? Like, for example, if you have like a plague or something like that.
If you have what? Sorry.
Build-up of some waste, and because I read a lot about the absorption, it's different depending on how is the health of your intestines.
Well, that's true of any organ, right? If an organ isn't as healthy, it's not going to function the way that it should, right? Mm-hmm. So its function will be lower. Mm-hmm. We're going to see how the digestive system actually adjusts itself based off of the nutrients that we add to it. So it's a little more complicated than just focusing on the small intestine and how well it can absorb stuff. We have to make sure that stuff is broken down properly while it's in the small intestine, because if we break it down too late, we're not going to have time to absorb stuff. And then the body's done a lot of work for nothing, right? Because it's used all of this energy to make these secretions that it's adding to this tubing. The tubing's contracting to force the food through it. It's a lot of work. So if we don't get all of the nutrients that we can out of it, it's kind of a waste. So when we talk about secretions being added to places like, say, the small intestine, we're going to see that happening right at the start, because that gives us time to mix it up well with the food stuff so that we get it properly broken down and can absorb stuff before the food leaves and goes to the large intestine. Clearly, the body can't use all of the food stuff that we intake. So whatever's left over that we can't use is removed from the body through defecation. And we're going to see that there are two sphincters in the anus, like we mentioned in lab last time, that are going to help to control that process. I like this figure from your book because it color codes all 6 of our processes. So notice we've got ingestion with kind of a light blue color, mechanical digestion with the darker blue. The yellow droplets and purple arrows are demonstrating chemical digestion. Propulsion is that green squiggly line. Notice propulsion happens through the entire GI tract. Where's most of the absorption happening? In the small intestine, just like we said, right? Because we see most of those orange arrows leaving from the small intestine. There are some that leave from the large intestine. Make a note for yourselves that we should also be seeing like just one arrow coming out of the stomach, because we do see some water and also some medications absorbed from the stomach. Now, ingestion and defecation, notice only have one arrow because they're only happening in one place. Mechanical digestion and chemical digestion are mostly right on this figure, but this is another thing that we should probably make a note about. It's not totally right because we said mechanical digestion was what again? That physical change that's kind of mashing up the food, right? And they're showing us that happening with these blue circular arrows. Notice we have some in the stomach and we have some in the small intestine, right? But where else do we talk about mechanical digestion? In the mouth, the oral cavity. So we should be seeing those circles for mechanical digestion there as well. Notice it's a similar thing for the chemical digestion. They're showing us chemical digestion happening in the stomach and the small intestine. But we know that there has to be some chemical digestion happening in the oral cavity as well because salivary glands are some of our accessory organs, and they're going to be secreting saliva that has some enzymes that you're going to have to know. So you might want to make this note for yourselves now that mechanical and chemical digestion are also going to happen in the oral cavity, even though it doesn't show it in this figure. So, since we've introduced these processes that help us with digestion, it's a good time to talk a little more in depth about that chemical digestion. We'll make a few notes about absorption as we do this, but we'll be talking about that a little bit more later. So we already know chemical digestion is going to be enzymatic. We're going to need some enzymes to help us break up these molecules, right, that are the food stuff. We could also classify the reactions that happen here as being catabolic. Do you guys know what that means? That literally means metabolism that breaks things down. What's the opposite of catabolism? Anabolism. Very good. Very good. So yeah, we're focusing on catabolism here. And what we're going to be focusing on is macromolecules getting broken down. So remember, when you learned about the macromolecules, you should have learned that they're polymers. What does that prefix poly mean? It means many, exactly. Many because they have many repeating subunits that are usually pretty similar, right? And those subunits we call monomers. What does mono mean? It means one. So that's telling you that that's one subunit. And remember why this needs to happen. Because the body cells can't take in these large polymers. We have to break them down to the monomers because those are small enough to pass through the cells. We could also classify these kinds of reactions as hydrolytic or hydrolysis reactions. So notice what I'm showing you here with this figure. We've got a polymer at the top of the screen, and each purple circle is representing one monomeric unit. If we decide that we want to remove one of those monomers, notice what we have to use. We have to use water to help us stabilize the ends that we just cut apart. So we split water, that's why it's called hydrolysis, right? Because hydro means water, lysis means splitting, so that it can cap off the ends of the new molecules that we just created. You guys with me so far? Okay. Questions? So let's think way back to general biology. What are our four macromolecules? We've got carbohydrates, which are the same thing as what? Sugars. Very good. What else? We've got proteins. Good. What was the monomeric subunit of a protein? Amino acids, exactly. And when we tether those amino acids to each other, when we bond them together, do you guys remember what kind of bonds we use? Peptide bonds. Very good. Why am I talking about this stuff? Well, because when we start to look at some of the enzyme names that help us with this, we're going to see some of these words in those names. All right, so we've got carbs, we've got proteins. What else have we got? We've got lipids, which are the same thing as what? Fats. Very good. And what's that last one that everybody wants to forget? Starts with an N. Nucleic acids. Very good. What are nucleic acids? DNA and RNA. And before we look really more in depth into all of these things, you know 1 out of the 4 has some properties that are different from everybody else. What am I talking about? What about the lipids? They're hydrophobic. Everything else is pretty much hydrophilic, right? So we've got to keep that in mind because that means we're going to have to treat those fats differently than everybody else. That also means that they get absorbed differently than everybody else too. So we talked about those. Let's look at these guys more in depth. We classify carbohydrates based off of their size. Regardless of the classification, we are always going to see saccharide as part of the term. So notice we have mono, which we know means what? One. Saccharide means sugar. So these are our simple sugars or our monomeric units because they only have one sugar group. It is important that you know that glucose, galactose, and fructose are all monosaccharides because this is what we hope to break down the carbohydrates to so that we can bring them in the body. If we stick 2 of these guys together, we form disaccharides because di means 2. And I do want you to know about sucrose, lactose, and maltose. When we talk about the nutrition and metabolism chapter, we're going to see more about where these things come from. But just to give you a little understanding now, table sugar, like what you might add to your coffee, is sucrose. Lactose we find in milk-based products, so like yogurt and cheese and milk. Maltose we find in anything made from grains, so like cereal, granola bars, beer, all of those things have maltose. The largest of the carbohydrates are polysaccharides, so that's clearly our polymer, right? Because it's got poly in that name. And there's only one that you need to know because it's the most common that we intake. Starch. So that's why your book really emphasizes that one. You do not have to memorize this entire figure. Okay, you don't have to memorize the whole thing. But I want you to focus right at the top because notice right at the top we're looking at the lumen, the open space of the small intestine. And we can see that we have our polysaccharide there, starch, right? And we can see that that is being broken down with the use of enzymes. How do you know that that thing that I put a box around is an enzyme? It ends in ASE, exactly. So notice how starch is being systematically broken down. We don't just take starch and turn it immediately into monosaccharides. First, we break it off in pieces to form disaccharides. And then we break those up further to help us make the monosaccharides that can now be pumped or moved into the cells of the small intestine so that we can move them across to the bloodstream. Any questions about that point? Pretty straightforward? Mm-hmm. Yeah? All right. Let's keep on moving. So you do need to know the names of the enzymes here, but before you freak out, let's look at those names. Notice 2 of them have amylase in the name. So that's one you're going to have to memorize. If you see amylase, it is an enzyme that helps us to break up polysaccharides like starch. And if we look at those names, they're also telling us who's making those enzymes, right? Who's making the first one? Salivary glands, exactly. And the second one's obviously being made by the pancreas, right? That's why it's being called a pancreatic amylase. And I already told you where those pancreatic secretions are going. Where are they going to be acting? In the small intestine. Good. The interesting thing is that the small intestine makes some of its own enzymes. And these enzymes are often referred to as the brush border enzymes because the cells of the small intestine have microvilli, those finger-like extensions. So because those microvilli are secreting the enzymes, they call it the brush border. If we look at the names of the enzymes that are made by the small intestine, well, they look an awful lot like some of those sugars that we were talking about, don't they? Like the disaccharides. We just replaced the O with an A so that we know we're talking about the enzyme now instead of the sugar. Right? Because we said lactose was obtained from milk-based products, maltose from grains, sucrose, table sugar. The book gives us a great summary table for this. You should know a lot of the stuff in this table. Don't worry about all of the wording here in the pathway of absorption. We're going to talk about that more generally, but the other stuff is important. So I know that's impossible to read, so let's take a closer look at carbohydrate digestion in that chart. So first of all, notice they're still showing us those same enzyme names. They give us a few extras. You don't have to know those. So I didn't talk about dextranase, right, on the previous slide. Even though it's in this table, you don't have to know it. I'm trying to go for the more straightforward ones. They're also showing us who makes those enzymes: salivary glands, pancreas, and small intestine's brush border, as well as where the enzymes are acting. And then they show us exactly what they're doing. So notice, both of our amylases are breaking up bigger carbohydrates like starch into smaller subunits like disaccharides. And then the brush border enzymes are going to help us to break up those disaccharides into the monosaccharides. And why was that again? Why was that happening in the small intestine? Because we have to get them to that size in order for them to be absorbed here in the small intestine. That's exactly right. So any questions about carbohydrate chemical breakdown? No? All right, then let's move on to proteins. We already know the monomeric unit is the amino acid. And we talked about those amino acids being bound together with peptide bonds. When they're bound together, those form polypeptide chains. But you guys should know that in order for those polypeptide chains to be true proteins, they need to gain a three-dimensional shape. You should have learned about that in general biology. We're not going to review that part here. But you should also know that proteins can be more than just one polypeptide chain with a three-dimensional shape. So remember when we studied hemoglobin, right? We studied it a couple times this semester. Hemoglobin had those 4 globin chains that are all stuck together, right? The globin chains were the polypeptide chains. So that's a specific example of a protein that's made of more than one polypeptide chain. They give us a nice figure for protein breakdown, kind of like they did for the carbohydrates. So notice once again, I'm not going to make you memorize every little thing here, but we start out at the same place, the lumen of the small intestine, and we can see that we have some polypeptide chains here that need to be broken up. And we've got some enzymes from the pancreas that are helping us to break them up into smaller chunks. We also have some from the brush border as well. When we break them up into the amino acids, then they need to move through a channel or a carrier, right, to get into the cell so that they can make their way across to the bloodstream. Pepsin is one of the proteins that, or one of the enzymes that you need to know that's going to help us break up proteins. So notice it's got pep in the name, just like peptide. Right? And notice where that's being made. That's being made in the stomach. Proteases are kind of a general term for enzymes that help us to break up proteins. If we look at the ones that come from the pancreas, you do need to memorize trypsin. Chymotrypsin still has that trypsin in the name, so that shouldn't be too hard as long as you memorize the trypsin. And carboxypeptidase, well, peptidase basically has peptide in the name, right? But we don't stop there because we also have some from the brush border from the small intestine. But notice, once again, they all have what in the name? Not just ace. Peptidase. They all have peptidase in the name, so they all basically have peptide in the name. If you know what a peptide is, you can read that term and know what those enzymes are breaking down. So if we go back to our chart, notice they're once again showing us the enzymes and who is making them. They're also showing us where those enzymes act. It shouldn't be surprising that the pancreatic enzymes are once again acting in the small intestine because that's where they're secreted to. And it's showing us a systematic breakdown once again. So pepsin isn't going to just turn our protein automatically into amino acids. It just breaks up the protein into bigger chunks. And then the pancreatic enzymes are going to help break those chunks up a little bit more. The brush border enzymes do our final breakdown once again, breaking down to our monomeric units. And I'll tell you guys right now, this stuff is a great place for an application kind of question. So if I told you that somebody ate a nice, lean hamburger, where would the chemical breakdown begin? The chemical breakdown would be in the stomach, right? We don't have any enzymes coming from the salivary glands here. Because if we're focusing on a hamburger, that's protein, right?
Yeah, but
in general, doesn't digestion start in the mouth?
you said, yeah. You said it.
Depends on what you focus on.
Chemical or mechanical.
Let's go back.
That's kind of tricky.
Mechanical.
It is. So you have to know the difference between chemical and mechanical. You also need to know where your enzymes are coming from. So like if we go back to carbohydrates, notice where their chemical breakdown begins. In the mouth, the oral cavity. So if instead I told you that somebody ate a donut for breakfast, where would that chemical breakdown begin? In the oral cavity. But the lean hamburger doesn't have any enzymes that are going to break it down in the oral cavity, right? That protein is going to be chemically broken down starting in the stomach. So try to think about those kinds of things as well. Try to apply what you know when you're eating your own meals. Lipids are another important one to talk about, so fats. We're only going to talk about one. Hooray! Triglyceride. Why? Because it's one of the more common ones that we ingest. You don't have to memorize the full chemical structure here, but I wanted to help you understand what triglyceride means. So this glycer in the name is telling us about the glycerol group that's part of the molecule. It also has a tri, which tri means what? 3. It means 3. So triglyceride has 3 of these fatty acid tails. It's only showing us 1 in the picture. But you should make a note that it technically has 1 glycerol group and then 3 fatty acid chains bound to it. And those act like the monomeric units for triglyceride. We already know lipids are hydrophobic, so we're going to have to treat them differently. What happens if you have a salad dressing that has oil and vinegar, and you shake those 2 things together? What happens? They separate, that's true, but when they separate, do they just form those 2 automatic layers when you shake them together? No, it looks like fat bubbles, right, or oil bubbles. Well, the same kind of thing happens in our GI tract when we eat a meal that's high in fat. The fat is going to separate from the water, so what it does is it forms these huge fat globules that are making their way through the GI tract. And that they only show you part of one up here. And that's a real problem because I told you that what kind of molecules are going to help us break down all of our nutrients? Enzymes. What kind of macromolecule is an enzyme? It's a protein. Proteins are usually what? Hydrophilic. Exactly, hydrophilic. So our proteins are in the secretions that are sitting outside of the big fat globule. What that means is that if we left the big fat globule stuck together like it is right now at the top of the screen, there wouldn't be very much surface area for our enzymes to act on. They could only act on the outer edge. There would be all that space in the middle that they wouldn't be able to get to. So we've got to break up our big fat globule, and that's what emulsification means. We're going to break it up into smaller fat droplets. And we do that with the use of bile, specifically salts that we find in the bile. And they're able to do this because they have something in common with phospholipids. We've talked about phospholipids previously when we talked about the plasma membrane. What are phospholipids like? All right, yeah, they have a head and they have tails. The head group is hydrophilic. Whereas the fatty acid tails are hydrophobic, right? I'm running out of room, otherwise I'd write the full term. So the bile salts are also going to have a side that's hydrophilic as well as a side that's hydrophobic. And because they like to interact with both fats then and water-based things, they're able to separate off little pieces of our big fat globule. And notice what that's doing. That's creating much more surface area for our enzymes to interact with at the edges now. We have less space in the middle that they can't get to, right? You guys with me? Okay, so emulsification is important. You should understand why it's happening here. we don't just need emulsification. We still have enzymes that we need to talk about. So shouldn't be surprising. Once again, we have an enzyme coming from the pancreas. And look at that name. It should be easy. It's got a lipid in the name, right? Lipase has a lipid in the name. When we think about how these substances, the lipids, get absorbed, are they going to need a carrier to help them out or a channel to help them out with getting across the plasma membrane of the cells? No. What are they going to do? They do simple diffusion. That's exactly right. They can just slip right through the plasma membrane because hydrophobic substances have an easier time getting through that layer with the fatty acid tails that are hydrophobic. So, when we look at our chart, notice it's still showing us the emulsification because of the bile that came from the liver, and it's still showing us the enzyme, the lipase from the pancreas, and it's showing us that, well, okay, the bile and the pancreatic lipase are both going to be acting in the small intestine, which we already know, right, because that's where the liver and the pancreas both secrete to. Not just to anywhere in the small intestine, but to where? What part? The duodenum. But they're showing us two other enzymes here. Luckily for us, they're both lipases. But notice one is acting in the oral cavity, the other is acting in the stomach. But the chart's telling us that they're of minor importance. Why would that be? Because what?
You still need the bile salts to break it down so that the lipase can get to it.
Exactly. The food, the lipid, hasn't been exposed to the bile salts yet at both of these 2 points. And we know the bile salts help to break up our big fat globules into smaller pieces so that the enzymes have an easier time acting on them, right? So lingual lipase and gastric lipase can only act on that outer edge of the big fat bubble, essentially, that's making its way through the alimentary canal. That's why they're of minor importance. So make a note about them. Know that they're of minor importance and why. Yeah?
I have a question about the example of the hamburger. It has proteins, but it also has carbohydrates and fats. So
Well, but whoa. So I didn't say the person ate the hamburger and the bun. I said the person ate a nice lean hamburger, right? So that would be that's the easiest example I can give you for a protein is talking about some lean meat, okay? It might not necessarily be a lean hamburger, but something lean. Whereas if I were to tell you that somebody ate a block of cheese, clearly that would be high in what? In fat and lipid, exactly.
Thank you.
You're welcome. Morgan, you had a question as well?
If we were given an application question for fats collected with cheese, would you want us to put the digestion begin in the small intestine and not in the mouth or stomach?
It would depend on how I worded the question. If I were to ask you where most of the digestion happened for the lipid, you wouldn't tell me the oral cavity then, would you? Because most of it's happening in the small intestine where we're emulsifying the fat and then exposing it to more enzymes. So if I ask where it technically begins, technically chemical breakdown does start in the oral cavity, but it's very limited. Anybody else have a question before we move on? Well, we're almost done with the chemistry stuff, so. Okay, we said nucleic acids were DNA and RNA. You should know that DNA is deoxyribonucleic acid, RNA is ribonucleic acid. They're named differently because they have different sugar subunits within their monomers, the nucleotides, ribose and deoxyribose. The enzymes that I want you to know here are super easy. Look at those names. What have they got in the names? Well, they both have nuclease. That's true, but notice we've got a pancreatic ribonuclease and deoxyribonuclease, right? So basically it has RNA and DNA in the name if you know what those stand for. Well, if they're pancreatic enzymes, where are they going to be acting again? Small intestine once again. Do make a note that there are some that come from the brush border, but I'm not going to make you memorize those names. So as long as you know about the ribonuclease and deoxyribonuclease for this one, you're good. We're going to go back to the overview of the digestive system. We have a couple more things to cover here, like how it's regulated and its circulation and layers of the wall. So when we look at regulation, it's our old friends, the mechanoreceptors and chemoreceptors, that are going to be helping us monitor things once again. What did the mechanoreceptors sense in our previous chapters? Stretch, because mechanical is physical change, and they help us to monitor stretch. And that's partially to make sure that stuff doesn't get overstretched, kind of like what we talked about in the lungs. Like we know the stomach has to expand and stretch as it fills with food, right? That's why we had to learn about the rugae in the stomach. But they're going to do more here. As the stomach stretches, that should tell the body that the stomach has food in it, right? And if the stomach has food in it, well, then it better be secreting stuff to its lumen to help break up that food. And it better be contracting the muscles in its wall to mechanically digest that food. Chemoreceptors. They sense chemical change, right? Is that going to be oxygen and carbon dioxide again? No. What do you think they'll monitor here? They will monitor pH because the stomach does have an acid, hydrochloric acid. Good. Anything else? They're going to monitor what kind of food we have in the organ. Because think about it, we have a bunch of enzymes that can be dumped into the small intestine. We don't want to waste our time by adding a bunch of amylases that break down carbs if we just ate that lean hamburger, right? Instead, the body needs to make more protein digesting enzymes. So that's all stuff that we talked about, except I didn't mention the osmolarity. We're going to talk about that more in depth later. That's going to be a really important thing when we hit the small intestine. For now, just make a note that osmolarity tells us about the concentration of particulates or solids within a solution. And like I said, we'll talk about that way more in depth when we hit the small intestine, because that's going to be a major issue for the small intestine. All right, and I already told you what they do, right? That they're going to help to control the secretions of things, as well as muscle contracting to help with things like mechanical digestion. When we look at how this control happens, so we know the receptors that are going to be monitoring things. We know what they monitor and generally speaking what that will do, but we don't know how. So we have intrinsic and extrinsic controls. Anytime we see intrinsic, that means that's a control that's happening from the system itself. So part of the system is controlling another part of the system. We see this a bit with, say, the small intestine. Small intestine is going to secrete some things that help to control other organs like the liver and the pancreas. Extrinsic controls come from other systems besides our current system. So our extrinsic controls, well, that's easy, right? That's always the central nervous system, and who else? What other system controls the body? The endocrine system, yeah. So we're not just going to be talking about nervous control. We also have to think about hormones helping with control too, right?
So would it be like the pancreatic enzyme that comes from the pancreas to the intestine?
No, we'll get to some of those specific hormones later. So notice I'm highlighting autonomic nerves here with the extrinsic controls. What were the two ends of the autonomic nervous system again? We have parasympathetic and sympathetic. Good. In our previous systems, like the cardiovascular system and the respiratory system, what did the sympathetic nervous system do? Yeah, it increased activity, right? Is it going to do the same thing here?
Yeah, but not.
No. No, it's going to slow things down. So you have to flip that around in your head. Remember, sympathetic does not always stimulate. Sometimes it inhibits and slows things down. Because if we're running from danger, right, because sympathetic's fight or flight, we don't have time to do things like stop and urinate. We don't have the blood going to the digestive system as much because we have to send it out to areas that need it more, like the skeletal muscle. So make that note for yourself. Sympathetic slows things down. Here, parasympathetic is going to be speeding things up. So those two areas are flipped now. And I already mentioned that we'll be seeing some hormones. We'll talk about them more later. Just make a note for now that we will be seeing some hormonal control. And we already talked about the autonomic nervous system stuff. I'm giving you this slide just in case you did not catch that. Our book gives us a figure to help illustrate both intrinsic and extrinsic controls. Notice, so we've got the intrinsic here, extrinsic here. With the intrinsic controls, our chemoreceptors are still going to be altering the nervous system in order to, they can move through two routes. They can either totally act locally or they can help with some of that extrinsic regulation by sending signals to the central nervous system so that the autonomic nervous system can modulate things. Another thing that they're showing us here is something we're going to talk about more later. Notice sight, smell, taste, external things, external changes can also change the digestive system. So like if you smell something that smells really good and you're hungry, you'll start to salivate, right? We see this with dogs, right? All the time. If you hold that treat and they really want it, the saliva will just be dripping out of their mouths, right? Well, that's because the organs prep themselves for the food before they ever actually see the food. And we'll be talking about that thought more next time. If we look at what the central nervous system does, notice we're seeing the effectors that we just talked about. We're going to see smooth muscle contraction changing as well as glandular secretion changing. We need to talk about serous membranes here, just like we have in every other system. Because think about it. In the abdominal cavity, where we find most of our digestive organs, we have a bunch of organs that are moving around in a space. So anytime that happens, we need serous membranes to make sure they don't cause friction with each other or with portions of themselves. Every time we talk about serous membranes, we change the name based off of where we are. So the general term for all of the serous membranes in the digestive system is peritoneum. But we can give that even more specific names depending upon where we find that peritoneum. So remember in lab last time we talked about things like the greater omentum and the lesser omentum. Those are specific examples of peritoneum. Okay, so if these are serous membranes, we know they have two layers. It shouldn't be surprising that we're going to see an outer layer that's called a parietal peritoneum. This is kind of a weird figure, so let's just make a quick note about what it is that we're exactly seeing. We have done a transection through the body, and we're looking down into where we cut. So when we look down into where we cut, notice there's this pink donut happening right here. That's one of our alimentary canal organs, like say one of the intestines or part of one of the intestines or something. So we have an outer orange layer of membrane and we have an inner one that's right in contact with the organ. What's that inner one going to be? That visceral peritoneum. And then there's that gray space in between the two. What's that? The peritoneal cavity. Very good. So that's all stuff you know. This other picture that I put in here, notice where it's at. It's below the liver, and it looks like it's hanging over the intestines, right? So that's the greater omentum. That's a specific example of parietal peritoneum that you've already studied. We just give it that more specific name because that's the peritoneum that's laying over top of the intestines. Now the tricky thing with the abdominal cavity being so big and all of these organs being in here together is that we're going to hit some points where the organs are not totally enclosed by serous membranes. When that happens, we call it mesentery. So that's where two layers of peritoneum are kind of coming up close to each other. They don't exactly touch each other though. There's going to be a little space there that's going to give us a route for things like blood vessels and nerves and lymphatic vessels to make their way through the abdominal cavity. So if we look back at the other part of my figure, where we've still done that same transection through the body, but we did it at a different point, we can see that now we have two organs from the digestive system. And notice there are points where what would have been visceral peritoneum are coming together. And there's a space in between, which is not the peritoneal cavity space. That's a space outside of the peritoneum. So that's a space for our blood vessels to go through, where I drew that little blue line and then just deleted it. You guys with me? Okay. We classify mesentery based off of location. What did dorsal mean? It meant the side by the back, the side by the spine, right? Side by the spine. Ventral is the opposite side, the side by the gut or the abdomen. Some organs will only be in contact with mesentery on one side. So we call these retroperitoneal. What does the prefix retro mean? Behind, like behind the times, right? So if it's retroperitoneal, It's an organ that's sitting behind the peritoneum. It's not enclosed in it. We see this a bit with some of the intestines. We also see this with the pancreas. Those that are surrounded are called intraperitoneal because intra means inside. And a good example of this is the stomach. Remember, the stomach had a bunch of peritoneal tissue associated with it.
What was the one that's inside? Intraperitoneal?
Intraperitoneal, yeah, it's this word over here. Which, if you know what the prefix intra means, that means inside, that's really easy. Okay, we're almost there, guys. We just have a couple more points to make. I want you to make a note that the circulation that's specific to these digestive system organs we call splanchnic circulation. And you actually studied part of this when you studied the blood vessels. Remember studying the hepatic portal system? That's part of this splanchnic circulation. And just as a reminder, remember some of those blood vessels were helping to carry blood from the small intestine, blood that had picked up nutrients from the small intestine, up to the liver so that the liver could process those nutrients, deciding if we needed to store them or modify them before sending them out to the rest of the body to use. The last thing that we need to talk about today is the layers of the wall, similar to what we've done with other systems. And put a big star by this point that I'm about to say. Next week, we are going to be looking at digestive histology in lab. Make sure that you review this information before then, or you're going to be lost that day because we're going to be looking at specific things in a lot of the organs. And if you don't know the general layers of the wall that I talk about today, it's going to be more confusing. Okay? So we have the same general layout, but we see it change a bit from organ to organ. So, the innermost layer that sits by the lumen is going to be called the submucosa. And as we work our way out, away from the lumen, we're going to hit the submucosa, followed by the muscularis externa, and then the serosa, which won't always necessarily be called serosa. Now, if the mucosa is sitting right there next to the lumen where the food stuff's going to be, what kind of tissue do we always have lining those kinds of spaces? So let me rephrase. You had 4 different classes of tissues. One of those classes is always aligning which class? Epithelium. Epithelium. And we're going to see that epithelium change just like we saw in the respiratory system. We talked about this a little bit in the respiratory system. In the oral cavity, oropharynx and laryngopharynx, what kind of epithelium do we have there? Stratified squamous. Stratified because it has many layers of cells, right? And that makes sense because at that point, the food has not been broken down much yet. Think about eating a granola bar or popcorn. That stuff that could damage the epithelium. And that would be really bad at these points that are so close to the outside of the body, these points that are exposed to more bacteria. It's true at the opposite end of the tube when we get to the rectum and the anus. We see stratified squamous epithelium again because we have a similar kind of effect. We're close to the outside and we see more friction. But would it make sense to have stratified squamous epithelium in the small intestine? No, because what happens in the small intestine? Absorption does, exactly. And we know if we're moving stuff across, we need a thinner layer. So through areas like the stomach and the small intestine and the large intestine, we see simple epithelium, but not simple squamous, simple columnar epithelium. Notice besides an epithelial sublayer, we're going to have lamina propria and muscularis mucosa. Haven't we talked about lamina propria before? What's lamina propria? Same thing as basement membrane or basal lamina. Good. What is that? Yeah, it's connective tissue that we always find by epithelial tissue because it acts like glue. It helps the epithelium to stick on to the other tissues near it. Muscularis mucosa, well, what kind of tissue is that probably going to be? Probably smooth muscle, that's right. Submucosa is going to be connective tissue. And notice that they're showing us a lot of tubing in that submucosa. So we have a lot of blood vessels, we have some nerves and some lymphatic vessels there. Really put a big star by that point that we find blood vessels in the submucosa because that's going to help you when you start to look at some of the slides next week. The submucosa can be hard to find, but if you see a blood vessel, you know that you've hit that sublayer or that major layer. Muscularis externa. What kind of tissue there? Muscle, mostly smooth muscle. But would it make sense to have smooth muscle all the way up by like the pharynx and the upper part of the esophagus? And the oral cavity? No. Because when we swallow food, that's a conscious thought, right? I've chewed my food enough that I can swallow it now. So you control that with skeletal muscle. Later, smooth muscle takes over. Similar deal at the other end of the tube. We'll take a closer look at the two sublayers of the muscularis externa in a minute. For now, make a note that there are two. And of the two, the circular layer is going to be inside of the longitudinal. So longitudinal is closer to the outside of the organ. If an organ has true serosa as its outermost layer, that's going to contain both epithelial and connective tissue. Because if it's serosa, it's a serous membrane on that outer edge, which means that there's a cavity outside of it. Okay, so when we think about what these different layers do, the mucosa is going to be a point for secreting a lot of things into the GI tract. One of the important things that we're going to talk about next week is mucus, which is going to have different functions. One of those functions is going to be acting as a lubricant. This is also where some of our enzymes come from, like if we think about those brush border enzymes in the small intestine, that was the epithelium of the small intestine secreting those things. Also thinking about the small intestine, it's going to be those cells that are in contact with the food that are going to be absorbing it. So it makes sense that our epithelium of the mucosa will be helping us once again. And we already talked about the fact that epithelium creates a barrier to prevent infections, like that stratified squamous epithelium that we're talking about in the oral cavity at upper portions of the esophagus. So epithelium is doing most of the functions here. Lamina propria doesn't help out a whole lot. It's really acting mostly like a glue. But muscularis mucosa is kind of interesting because it's got muscle there. And it's got muscle that we don't think about. That muscle is not the muscle that helps to push the food through the tubing. That muscle does small contractions, which we call a twitch. And that's important because if you think about some of that food stuff moving through the tubing, like going back to the granola bar or the popcorn, you can imagine that some of those little sharp pieces might get lodged in the sides of things like the esophagus. Well, it can't stay there because then it might cause a blockage or something, right, the next time we try to swallow. So the twitch of the muscularis mucosa helps to knock that stuff off the side of the wall and back into the lumen so we can keep it moving through the tube. We already mentioned that submucosa is going to hold things like blood vessels and nerves, so we're going to see some of our control happening here. We're also going to see nutrients and oxygen being provided to our organs here. Muscularis externa, we talked about the fact that this has two sublayers, right? A circular and a longitudinal layer. And that's why I'm giving you this picture on this slide, because this should help you to understand why we name those the way that we name them. Depending on the type of cut we do for the organ, we're going to see those two layers differently. If we do a cross section where we cut along the width of the tube, like what we're looking into right here where it looks like a donut, in that case, we're going to see the full cells in the circular layer because those cells are lined up end to end around the diameter of the organ. Notice when we do that same cross section, if we look at the longitudinal layer, well, that doesn't look like smooth muscle at all. It looks kind of like pebbles. Well, that's because those cells are lined up end to end along the length of the organ. So when we did that longitudinal cut, we cut through the middle of those cells, and now we're looking down into where we cut. If we were to do a longitudinal section and cut along the length of our organ, the cells would be just the opposite. We'd be able to see the full cells in the longitudinal layer, and then the circular layer would look like pebbles because we would have cut through those cells. Yep?
Just then the PowerPoint says differently because they say when you cut a cross section, it will show you the longitudinal one, and the longitudinal section will show you the circular.
Well, that's a typo because they're named based off of how the cells line up end to end. So if we do a cross section, we're going to see this kind of view with the longitudinal layer, but if we do a cross section, we're also going to see this kind of view with the circular layer. So thank you for making me aware of that. I'll make sure to correct that before I post the PowerPoint today. Do make a note that this is the layer that's responsible for moving the food and for mechanical digestion. So for propulsion and digestion. There are some modifications in it. We talked about some sphincters in lab last time. So when a sphincter happens, it happens in this major layer. Remember, that's just a ring of muscle that can do what? It can contract so much that it totally closes off the tube. So it acts kind of like a door, regulating what moves from what organ to the next. I told you that most of the time we're going to see serosa as the outermost major layer, but sometimes there will be adventitia instead. The difference is adventitia is just connective tissue. And really the only time we see an organ with only adventitia, well, that's the esophagus. And that should make sense if you think about the esophagus, because think about it, the esophagus does not always move through cavities. When it's in the neck, there's no cavity there. And because there's no cavity, we don't need serous membranes. Instead, we can just glue it onto the surrounding tissues. That's what adventitia does. Because it doesn't have anything to rub up against to cause friction. There's no space for that. So that's it for the layers of the wall. Next time we'll get into all of the parts and exactly what they do. So we're going to look at more functional anatomy next time. I can tell you guys are worn out. So if you have a question, come up and talk to me during your break. Otherwise, I'll see you guys back in here in about 10 minutes.